Next, let us learn about the pistil, then about the Megasporangia, that is the ovule, and embryo sac, that is a female gametophyte. So all these includes the female reproductive part of the flower, which is called as gynoecium. Okay, so this all includes or comes under the gynoecium, that is the female reproductive part of a flower. So let us learn the parts one by one. The female reproductive part is otherwise called as pistil. You might have come across the term pistil. Also, you might have come across the term carpel. So we must know what is the difference between pistil and carpel, or what do they signify, or what do they mean by the term pistil and carpel. In this figure, you can see this as a pistil and this they have shown as the carpel. So we have already learned how a pistil looks like, right? A pistil will have a stigma, a style, and an ovary. So this is what we call as a pistil: stigma, style, and ovary. So this region is called as stigma. The structure of stigma varies from one flower to another, and this lenter tube is called the style, and the basal swollen portion is called as the ovary. If you take the section of ovary, you can see regions like this, and this each segment or each chamber or each part is called as carpel. You understood? So this whole thing is actually the pistil, and the part inside the ovary, if you take the section of the ovary, you can see like this, and that part is called as the carpels. And in this figure, you can see that in this, the first figure, it has only one pistil, as well as if you take the section, cross section of it, you can see only one carpel. And in this case, you can see three pistils, and if you take the cross section of it, each would be separate carpel. And in the third figure, you can see that only one pistil with three different stigma or style and stigma, and the basal portion is only one region. But if you take the section, you can see three carpels, right? So the number of carpels vary from one flower to another, and I hope you understood what is the difference between a pistil and a carpel. What do they denote by the term pistil and carpel? In this case, there is only one carpel. So if there is only one carpel, we call that flower as monocarpellary. Okay, since they have only one carpel. In this flower, you can see it is more than one carpel. We call it as multicarpellary. So in a flower, if there is only one carpel, we call it as monocarpellary, and if they have many carpels, we call them as multicarpellary. And you can get now you get a question here. What do we call this one? In some cases, this multicarpellary, see this here in this also, the case is multicarpellary, because in a flower, there are many carpels. But if all the carpels are fused together, they are joined together, we call as syncarpous. This ovary is syncarpous ovary. Multicarpellary, syncarpous ovary, because they are fused. And here in this case, we call them as apocarpous ovary, since they are not fused, they are free, they are not joined. So these are multicarpellary apocarpous ovary, and this is multicarpellary syncarpous ovary. Now let's look at the figure given in our textbook. You can see the pistil, stigma, style, and ovary. And this is again another pistil, but in this case, you don't see the style, you see many stigma and a single ovary. Since they have many stigma, we understand that there are many ovaries, and all ovaries are fused. So they are syncarpous ovary, they are fused. And in this case, each one is different. You got it? And the actual figure of this mechelia, this is mechelia. Then actual picture of mechelia you can see here. See this yellow color part which you see here is actually the stigma, style, and you can see that the basal ovary. So in this flower has many ovaries, that is many carpels, and they are not joined together, they appear separately, and so we call it as apocarpous ovary. So syncarpous ovary and apocarpous ovary. Now what you see is a section of a fruit. Actually this fruit is developed from an ovary. So let us consider it as a section of an ovary. So in this section of an ovary, so these chambers, it is multicarpellary, okay. So this part This part is a carpel. This is another carpel. This is another carpel. This is another carpel, and this is another carpel. So that they are multicarpellary. And since you find here 5 carpels, we call it as pentacarpellary. And in this, you can see only 2 carpels, so they are bicarpellary ovary. And this both are syncarpous because they are joined or fused. So they are syncarpous, multicarpellary, syncarpous ovary. And you can see in this, the small minute structures are called as the ovule. So here in this case, it is already developed into a seed, but let us consider it as an ovule. Okay, these minute structures are called ovules, and inside the carpel, these ovules are arranged in a chamber. That chamber is called as locule. Okay, so this is a locule here, and inside the locule, the ovules are arranged. And you can observe that these ovules are attached to the carpel or the ovary with the help of a special kind of tissue, and that tissue is called as placenta. So kindly recall the placentation and types of ovary which we have studied based on placentation. So this is actually axile placentation, and here also it is axial placentation. You can see here clearly the placenta is clearly visible here. So the placenta is a structure which helps the ovule to get attached to the ovary. Okay, and now we can speak in the terms of ovule. This is actually seed. For your understanding, we have put this figure. So let us talk in terms of ovule. So ovule develops or starts from this placenta. So placenta will be like this, and on that the ovule will be developing inside an ovary. So this is how the development occurs. So now let's talk about the development of ovule or the megasporangia. So the ovule is the megasporangia. Okay, and this is the ovary. Inside the ovary, here there are many megasporangia. Let us study about single megasporangium. Also, you can observe here the number of ovules differ. The number of ovules vary from one plant to another. For example, if you take the examples given in your textbook are wheat, paddy, mango, in all those cases only one ovule is found, or only one seed you find, right? So only one ovule. Whereas in papaya, watermelon, you find numerous seeds, so numerous ovules. Let's see the structure of megasporangium and how does it develop. So this is the structure of a typical ovule of an angiosperm. Okay, so this structure, you can see it is clearly marked as ovary. So this is ovary wall, and here comes the placenta, and this placenta is attached to the ovule, that is the megasporangia, with the help of a stalk, and that stalk is called as funicle or funiculus. In your textbook it is given as funicle or can be termed as funiculus. So funicle is the stalk by which the placenta is attached to the ovule, or ovule is attached to the placenta. It is the stalk. And the funnel shaped region where the funicle attached to the ovule, and this region is called as hilum. This region is called as hilum. And now this hilum, it represents the junction between the ovule and funicle. You can see each ovule is protected by integuments. The covering is called as the integuments. Integuments are the covering, and there are two coverings here, two integuments here, and inside the integuments encircle, or inside the integument is the nucellus. It's a tissue, and the integuments are not joined. They leave an opening at one region, and that opening is called as micropyle. You can see the micropyle end here, a micropyle there, and the region opposite to micropyle. In an ovule, the region opposite to micropyle is called as chalaza. So if the micropylar end is here, and the region opposite to micropyle would be chalaza. So this is a typical structure of an ovary with ovule. You can see that style starts from here. So it appears like the style and ovary. Now let us compare this diagram to the diagram in our textbook. Now this is the diagram given in our textbook. So you might find a little difference, a small bit of difference here, because it is inverted, right? You can see it's upside down. So you can see this is the funicle. So we learned funicle here. and this end is micropyle. So the region opposite to micropyle is chalaza. So this is the chalaza end, chalaza pole, and the region opposite to the micropyle, and you can see the two integuments, inner and outer integument, and inside this tissue is the nucellar tissue. So we did not learn about the embryo sac. We are talking only about the megasporangium. You know, megasporangium will be holding the megaspore. And this kind of ovule, this kind of inverted ovule, so it is inverted like this, and this kind of inverted ovule is called as anatropous ovule. So only this term is given in your textbook as anatropous ovule. So when we mention this anatropous ovule, you must know about the other positions of ovule. So this name we gave based on the position. So let's see the other positions also. Now you can see the other types of ovule. Atropous ovule. So this typical structure which we have learned is actually atropous ovule. That is a straight one. And this is anatropous ovule. What we learn here, this one. So also don't confuse this anatropous ovule. They have shown like this in our textbook. In your question paper, you may get like this also. This anatropous can be drawn like this too. Okay, it can be drawn like this. So here the integuments and the micropyle come on the, here the integuments and the micropyle come at this end. It can be drawn like this. So this both are anatropous ovule. Amphitropous, that is partially, campylotropous, hemianatropous, cercinotropous. So based on, you can see how the ovule is arranged. Based on the position of the ovule, it is classified into many types. And we are learning the anatropous ovule, that is the inverted one, inverted type. Okay. So we know that ovule is the megasporangium. Inside the megasporangium will be the megaspore. So let's see how that megaspore is formed inside the megasporangium. Now let us talk about megasporogenesis. Megasporogenesis, that is the formation of megaspore. This is a megaspore. You can see that this is the ovule, where this is the micropylar end, and inside this tissue is nucellus. And generally in angiosperms, generally megaspore develops from a single megaspore mother cell. The whole nucellus, inside the whole nucellus, one cell will be differentiating as the megaspore mother cell. You can see here one cell. This one single cell will be differentiating as megaspore mother cell, MMC. That will be towards the micropylar end. One cell in the nucellus will differentiate as megaspore mother cell. And that cell will be having a dense cytoplasm with a prominent nucleus. And this megaspore mother cell will undergo meiotic division. Okay, it undergoes meiosis. So, currently it is in diploid state. Now it undergoes meiotic division. First it divides into two. The first meiotic division, it forms a dyad. It divides into two, and then it divides into 4. So once the meiotic division is completed, we get megaspore tetrad, and each haploid in number. Out of those 4 megaspores, so megaspore mother cell divided meiotically to form megaspore tetrad. So out of this 4, only 1 will be functional and all the other 3 will degenerate. Only 1 will be functional. So this one represents that functional cell. Okay, so functional cell with 1 nucleus. Now what happens is the nucleus of this functional megaspore. So this is one megaspore, one functional megaspore is undergoing mitotic division. So what you see here is mitotic division. So it is dividing mitotically and the nucleus is dividing into 2. So it is 2 nucleated stage. And followed by, you can see that each nucleus is again dividing into 2. First, one nucleus divided into 2 and each nucleus of that is again dividing into 2. So 4 nucleated stage, and now you can see here as 8 nucleated stage. And one of the significance or prominent or uniqueness of this is here the nuclear division is not followed by cytoplasmic division. So that's why we get 8 nucleated stage. So once if the nuclear division is followed by the cytoplasmic division, each nucleus would be surrounded by a cytoplasm and it should be a single cell. But in this case, it's not like that. It is not followed by the cytoplasmic division. So finally, it reaches the 8 nucleated stage. This 8 nucleated staged cell, so this is one cell, right? So what happens is, each one starts getting or attaining or gets the cytoplasm and differentiates into cells. So always remember, they always mention this is the micropylar end, okay? This micropylar end, 3 cells there. So imagine this is a structure, and now this structure is called embryo sac, because embryo is gonna grow inside this. So we call it as embryo sac. Embryo sac is in 8 nucleated stage. Out of this 8 nucleus, 2 nuclei will be moving towards the center and other 3 nuclei will be on each side. So this side is marked as the micropylar end. So the nuclei at the micropylar end will develop cytoplasm around it, and out of the 3, 2 will be developing as cells called synergids, and one of it will form the egg. 2 will be developing into synergids and 1 will be differentiating into egg. And then the cells at the chalazal end. So this is the chalazal end, the end opposite to the micropyle. We call it the chalazal end or chalazal pole. The 3 nuclei at the chalazal end will form or differentiate into 3 cells called as antipodals. And what happens to the cell at the center, nuclei at the center? This both nuclei at the center will form a central cell with 2 nuclei in it. So this is central cell with 2 nuclei in it, and those 2 nuclei are called as polar nuclei. They remain as 2 different nuclei. So this 8 nucleated stage, there are 8 nuclei here, but only 7 cells are there. 7 celled stage, because the central cell has 2 nuclei, 2 polar nuclei, and other 6 different cells. So 8 nucleated, 7 celled stage is called as embryo sac. And this embryo sac is the female gametophyte, which is carrying the female gamete egg cell. So now we have learned the transformation from Megasporangium to female gametophyte, which is carrying the female gamete. So this is the fully developed embryo sac or female gametophyte with all 7 cells and 8 nuclei. You can see here they have shown the chalazal end here. Remember, whenever the figure is flipped upside down or anywhere, you must always note the chalazal end and the micropylar end. The chalazal end carries the antipodal. So this is the central cell with the polar nuclei, and here the synergids at the micropylar end with egg. And in the synergids, you can see a special structure called filiform apparatus. So we will be talking about the significance or the function of this filiform apparatus.